COCA for inviting me here. Really, I've always found that COCA is a phenomenal group, and I know I'm preaching to the choir here, but this really does mean a lot to me as much as I know it means to you. So it's really an honor to be invited to come talk to you. So again, I want to point out that it is really important to know who runs the clinical trials when we're talking about this. And this is, again, broad strokes. It's important to know the basics of trial design and running a trial to understand why you as the individual are getting benefit, but also how we're improving the science and future of all of cancer care. So currently the NCCN, which is the National Cancer Center Network, says that all patients should be offered clinical trial if available to them. And my goals of this talk are really to provide you with a better understanding of trial design and implementation so that you understand why and what we're doing together amongst patients, providers, and caregivers. Please appreciate that this is a 30,000-foot view and that each of these topics could be its own hour-long talk. So who runs the clinical trials is really split between three major groups: the government, industry, and when I say industry, I mean pharmaceutical companies, and then investigators, and those are people like myself. And that really is a complex web of who runs these. It's never just one individual group. There's really complex connections between all of them. And there are a number of consortiums that people like myself belong to, such as things called the NRG, the GOG, SWOG, et cetera. And these are all brought together to bring the trials that we know of. So diving into each of these groups, the government is split up into, obviously, is a complex web. It starts off with the NIH, the National Institute of Health. That breaks down into the National Cancer Institute. And then there's subdivisions of that. We call them CTEP or ETCTN. And those are some of the most prevalent ones for GYN cancers. And so what are those? So CTEP, as we refer to, is called the Cancer Therapy Evaluation Program. And they're a network that we work mostly with GYN cancers. And they work to either solicit trials from us as investigators, or we solicit trials through them. They're really hoping to be a bridge between us and the pharmaceutical companies to help design and run trials. And so they even have groups of something that we call the CRADA that they work with certain pharmaceutical companies to allow use of certain drugs. And there's a whole list of them that we can help formulate trials with. And that helps curtail things like costs and bureaucracy and things like that. Now, obviously, the pharmaceutical companies have say in all of that, but it really makes this streamline a lot more seamless between those three individuals, meaning industry, government, and trialists. Now, industry, so that is, as we call, the pharmaceutical companies. And I know, especially now that we have pharmaceutical companies really in the headlines with all the trials that we've all been talking about. And I know there's been great buzzwords like phase 1 trials and everything that in the New York Times and all the everything with the vaccines. And unfortunately, there's been a lot of negative press towards pharmaceutical companies about this. And I say it's unfortunate because it really points a one-sided picture. My perspective of this is that in order to advance science and advance ultimately patient care, we really have to collaborate with everyone. And those three groups that I talked about are really a true collaboration. Pharmaceutical companies, yes, they are for-profit companies, but they want and need to work with us to get to that. We as the providers want to do the best things that we can for people, and the government also, same thing, they want to provide the best level of care. And while sometimes it's true that cost dictates a lot of things, the reality is at these stages of things, it's not those aspects that are driving things. It's my belief that everyone that I communicate with and is really out to help improve patient care and really try to cure cancer. We would all love it if we were out of a job. And the truth is that these clinical trials are not money makers. Nobody's making money off these clinical trials. Yes, they are hoping to get approval for drugs, and that's where these trials are running towards. But at this stage of the game, these aren't considered money makers. So, I don't want people to ever think that that's what's kind of driving all this behind. And then investigators, so people like myself, we're always allowed to propose things called investigator-initiated trials, and that's to any of these consortiums. And what we do is we submit them and try to point out mutual relationships between us groups, and hopefully try and find ultimately funding to help get these ideas that we have out and move the science, what we call move the needle forward, to improve patient care. So clinical trial phases. So fortunately, we've seen a lot of these buzzwords in the papers recently with the vaccine trials. And I want to kind of better clarify what these phases of trials are. So as I said before, the NCCN has a broad spectrum statement that says all patients should be offered clinical trial when available to them. But that doesn't dictate what kind of clinical trial and what do we consider from a trial standpoint is appropriate for anybody. And that really tailors into the patient's really specific history. And except for, we always favor later phase trials. So I'm going to go through what these phases really mean to help you better understand that. And so here I show that on the far left side, we have preclinical trials. And those are trials on cells or animals, usually mice. And they're to really help dictate what dosing may be appropriate, what combinations may be appropriate, how the effects are for cancer types to move into trials before offering them to humans. And that gets into phase 1 trials. These are first in human trials. Phase 1 trials usually have a low number of patients. They still may be national level trials, but their goals are more for safety rather than efficacy. Now, that being said, this is something that some of you may have heard me say. If I'm presenting you as a patient of mine a phase 1 clinical trial, my goal and your goal aren't to see if it's safe in you. I would never provide you with a clinical trial option that I didn't think had a rationale for efficacy for you as an individual. And we'll get into that in a little bit more detail later when I talk more specifically about phase 1 trials. Phase 2 trials then have a larger number of patients, and their real goal is for efficacy. And so to prove statistically that they work in a certain patient population. And then phase 3 is a larger confirmatory population of thousands of patients, and it's typically comparing towards the standard of care. And then phase 4 trials, these are less common because they're typically already FDA approved drugs. They're really looking for response and long-term safety efficacy in a whole population base. So we'll probably see phase 4 trials for the COVID vaccine after a couple of years and after it's been out, when we've really provided it to a whole worldwide population standpoint. But they're not common to see in a cancer setting. So for trial design, as you can imagine, moving through those phases to get through to FDA approval really historically has taken quite a bit of time. Back in the 1980s, 1990s, early 2000s, this drug development really took on the order of a decade. I think at one point, the average time from development into approval was on the order of 9 to 10 years. And that has become less sustainable and really isn't an option anymore. The reason is because science has moved so far forward. Our science, as far as things like picking up biomarkers or assessments, is moving so rapidly, and our knowledge about cancers is moving so rapidly that by the time something got approved on that time frame, we're already years ahead from further drug development. And so it doesn't really make sense. So as you can imagine, as I stated, these things take more time, but they also take significantly amount of money. And so nowadays, clinical trials are less designed to go through phase 1, then stop, then phase 2, then stop, then phase 3. They're more congruent to try and help move things directly to FDA approval. So we've seen some combinations of drugs approved off of phase 2 trials. We've seen some design of trials designed as one big lump package, so moving phase 1, phase 2, and phase 3 into one large trial. Lots of different mechanisms to try and help shrink down this time frame, as well as the funding to help get these done more quickly. So let's dive into the different phases for a little bit more specific, so you kind of have a better understanding when these things are presented to you or people that you know. So as I said, phase 1 clinical trials are really designed around safety. The main goal of these is to determine something that we call the MTD. That's the maximum tolerated dose. And as I said, these are first in human drugs or first in human combinations of drugs. And the second aspect of phase 1 trials is to evaluate the safety of these. And there's a number of different mechanisms that we do this, but the classic that we do it is something called a 3x3 design. And that means that we start off with 3 patients at a certain dose, and we see if anybody has something called dose-limiting toxicity. So we're looking at all the safeties, but depending on the drug, we already know quite a bit about the possible toxicities before we've even started a trial. We try to cater towards the expected toxicities and unknown toxicities. And if nobody experiences any toxicities, then we move up a dose. And there's already a prescribed course into moving up. And then we move up until we find that a certain number of patients really can't tolerate the drug or have had certain side effects that we were expecting. And then we drop down to what we call the maximum tolerated dose. And that's the kind of classic way that we do phase 1 trials. So there are very rarely and really never placebos in phase 1 trials, because we want to know exactly what you're getting so we can determine, is it safe and what's the right dosing. Now, I've stated that that's classic 3x3 design is the way we've run phase 1 trials, but there are many different ways to actually design these phase 1 trials. As I said before, we typically did 3 patients at one dose, then 3 patients at the next dose, but there are other ways for different reasons to do that. We can accelerate the dose in a single individual, meaning increase the dose on that individual until they have a side effect. So there's obviously drawbacks and benefits to both. And again, these are only 2 examples of more than 30 different ways to design a phase 1 clinical trial. But I give these as examples, and it's always comfortable to ask your provider, or if this is a consideration for you, kind of what you'd be getting, because again, in phase 1 trials, we know exactly what you're getting and we know why, and so we'd be able to give you that information. Now, one of the popular questions that I get in my phase 1 clinic is, why would I ever go on to a phase 1 trial? You just told me it's about safety and maximum tolerated doses. How does that benefit me as the patient? And this is actually a classic concern that dates back to even what we concerned as were phase 1 trials ethical to give to people. And this was a very hot topic a couple decades ago of the ethics of clinical trial. And since that time, we've really shown that there is significant safety and ethics involved in these early phase clinical trials. And what I found is that there was a lot of research into these in other tumor types, but nobody had ever looked at it in ovarian cancer. So this past year, I actually published an article on looking at specifically survival and outcomes in phase 1 clinical trials for ovarian cancer patients. And this is all patients that we've treated at our hospital, University of Colorado, over the past 10 years. We really found a couple very interesting things. First, we found that the majority of people actually were on multiple phase 1 trials, which means that they may have been on a trial that it worked for a certain amount of time, and then it stopped working, and then they went on more phase 1 clinical trials. We, as expected, found that the majority of people had a number of different lines of therapy before. So the average was 5 to 7 lines of prior chemotherapy regimens before going on to a phase 1 clinical trial. And here's where we looked a little bit at efficacy, because people, rightly so, want to know, is this going to work for me? And what we found is that we were able to curtail the whole safety issue. We found that no patients over 10 years died from a toxicity from any kind of drug that we were giving in a phase 1 clinical trial. Only 2 patients actually passed away on trial because of disease, and that the response rates were extremely comparable to standard of line therapies, meaning that if you weren't on clinical trial and you went with whatever your provider recommended as a standard therapy, they were relatively equivalent. Now, ORR stands for response rate. And when we measure response rates, we're looking at, is there tumor regression or complete response? And while 15% sounds low, remember that this is a heavily pretreated patient. So this is a patient who's been through 5 to 7 lines, even more, of therapy before. And so response rates are generally going to be lower. But we found that the clinical benefit rate was close to 50%. And what do I mean by clinical benefit rate? That means that we were able to either shrink the disease or keep it stable. And many of you have heard me say this in the past, that at that stage of the game, unfortunately, in a non-curable disease, keeping the disease stable from growing or shrinking it a bit is a win in my book. And we were able to show that that was pretty significant. What I really want, I'm going to go back a second, because what I really wanted to also show in this trial that I think we did is that historically people have thought of phase 1 trials as, well, let's go to phase 1 trial when there's nothing left for me to offer you, or when there's no options left. I've heard this term a lot. And I want to stress that I don't think phase 1 trials are a no options left option. I think that these are really viable options that can help patients for their specific tumors. So moving on to phase 2 trials. And as I said, that phase 2 trials are looking now at efficacy. So we've proven their safety, and we have proven what the correct dose is. And now we want to statistically prove that it works in a certain patient population. And there's a number of different ways to do that. So we continue to evaluate safety because it's a larger number of patients. But then we need to compare it to something, right? If we want to prove it's efficacious, we want to compare it to either a placebo to show that it's better than a sugar pill, or historical control. And historical controls mean that we are comparing it to a patient population and what we expect the response to be based on prior therapies. These are getting harder to do in modern times because we have a number of more patients on clinical trial. We've had a number of patients be on therapies that didn't exist, even 2 years ago, let alone 5, 10 years ago. And so historical controls are a little bit harder these days to do. And so a lot of phase 2 trials do have placebo. And that's always told to the patient right away that, there's X percentage chance of getting a placebo. Sometimes it's 50-50, sometimes it's 75-25. It's all depending on how the trial is designed. And we won't know if you're getting the placebo or not, but we will know the rate of whether we're randomizing, in a 50-50 range or different. So you can take that into consideration of whether this trial is right for you. So moving on to phase 3 trials. Phase 3 trials are now the larger thousands of patients across the country or world, and trying to prove that it's better or equivalent to the standard of care. Now I point these terms out: superiority, non-inferiority, and equivalence. Phase 3 trials may or may not include a placebo, but they always have a couple different arms on the trials, at least 2, sometimes more. And what they're trying to look at is a specific question that's designed by whoever designed the trial. Now, superiority trials are looking to say that this new regimen is better than the other one. Non-inferiority trials are trying to show that it's no worse than this other therapy. Now, the majority of phase 3 trials these days are designed as non-inferiority trials. And why is that? You may want to say, well, we always want to make something better, right? It's not always that simple. Sometimes we have a new combination that we think is actually equivalent, but is significantly less toxic, meaning a lot less side effects, or it could be a lot less financially toxic, so a lot less expensive to the individual. And so we would rather show that this is not worse than that other line of therapy. And it's different than equivalence, because non-inferiority doesn't really show that they're the exact same or they're equivalent, but it means it shows that it's no worse. All this gets very complicated into statistical analysis. And trust me, I am no statistician. I always have at least one statistician on any trial that I'm designing, because it gets way above my head too. But the pitfalls of trying to do something like a superiority trial is if you don't prove that your new drug or drug regimen is not superior, you haven't proven that it's not non-inferior. And so that actually can ruin a whole trial and all the information you gain from it, because you weren't able to prove how you designed the trial. And non-inferiority patients may require more patience from a statistical standpoint, but can typically be more beneficial in the long run. So this gets a little bit into the statistical design of how we think of things, of how we need to design things, and that gets into power analysis and lots of statistical things that I'm not going to spend a lot of time on tonight. But ultimately, it goes into how we actually design the trial to make sure that our goal is to always have a positive trial, to prove our hypothesis. Or disprove the opposite of the hypothesis. And these are things that we're all considering in trial design. So eligibility, this is always a hot topic, because you may have heard me say, or my colleagues say, or are you eligible for the trial? So it's not uncommon for me to call you up and say, I've got a trial I'm thinking about for you. I need to check if you're eligible. Well, obviously I want you to be eligible, because I want you to be on the trial. But we have to go through things like screening and inclusion and exclusion criteria. Now, why do we design the things like this? So screening means that we've set criteria of who we're looking for to be on this trial. And that ranges for a number of different reasons, but I can tell you there is a significant amount of thought process that goes into designing these aspects of it. The most basic of this is considering validity versus feasibility. And so what I mean by that is, if you designed a trial that was all inclusive, then there's too much variation in the population that you're studying, and your validity may go down because of that. On the counter, if you are too exclusive and you don't include enough people, or aspects, then you're too exclusive and you can't enroll in the trial, and you just can't get any patients on trial, and that's feasibility. And these are the things that we counterbalance amongst them to get people to get the right patient population for specific reasons for each individual trial.
Dr. Corr, it's Janine. I'm going to pop in with my own question here. Great. Something that's always kind of puzzled me. I know that ovarian cancer is technically a rare disease, and there's not a very large population of women that we can even get data from, basically. How can you address how that impacts clinical trial, structure? Absolutely.
So this gets a bit more into the statistical design of things. You're right that ovarian cancer is a rare disease when we're talking about a patient population, right? The overall incidence is 1.7% of women, and that is rare. The reality is, from a ovarian cancer standpoint, when we talk about certain histologies, so the most common being what we call high-grade serous carcinoma, That type, we can design trials really without significant statistical manipulation, because on a national level, there are enough patients to prove or disprove the ideas that we have. It's uncommon to have phase 2 or phase 3 trials at a single institution. That's why we have these consortiums that we do. We open these trials a lot across many different sites across the nation in order to enroll and get those numbers that we need. However, there are rare type ovarian cancers, just like any other types, that are even rarer amongst rare. And those are typically, we consider different histologies, such as low-grade ovarian cancers or mucinous ovarian cancers. And these are much harder to, and we struggle with what the appropriate way to study these is because in the classical sense, we cannot ever power a study to analyze to compare one arm versus the other. So these are where we very much implore that historical control aspect that we do to try and see if it's better or not. And that's how we typically get things like FDA approval in the rare of rare tumors. Because there are certain rare tumors that even the government, the FDA, acknowledge we will never compare, let's just say, carboplatin and Taxol chemotherapy versus agent X in a mucinous type cancer. You know, this just even nationally don't get the numbers that we need to prove one is non-inferior or superior to the other. And so those considerations are taken into fact by everyone involved.
Thank you. That helps a lot.
Yeah, thank you. That's a great segue into kind of one of the other considerations of clinical trials, randomization, and how do we do those power analyses to really prove one is better or non-inferior to the other. Randomization is what you may have heard of, one of our gold standards, right? We always want to have not a randomized clinical trial. And what's the purpose between randomization? And the simplicity of randomization is that we literally have a formula that a certain number of people will get one arm of the trial and a certain number of people will get the other arm of the trial. But we want to ensure that those divisions are split equally. And we decide who gets randomized into what. And what I mean by that is we don't decide that you're going to go into this arm or that arm, but we decide that a certain percentage of patients with BRCA mutation are going to be on each arm of the trial, or a certain number of high-grade serous ovarian cancers are going to be on each number of the trial. And what we do is we do that so that we make sure that randomization is equal. And so if you think about it as simplistic as age, we want to make sure that everyone in one arm of the trial wasn't 90 or older and everyone on the other trial was 40 or younger, because obviously that's going to affect how your results are. So we think of randomization as a way of eliminating bias. And there are biases that are intentional, but there are many biases that are non-intentional. And a randomization really helps remove those. The thing to remember is that randomization does not remove all bias, though. You can never remove all bias. So there are biases in even just trial design. So things like, as I talked about before, if I designed a trial for non-inferiority versus superiority, there's a bias that I've implemented in there in how I've designed the trial. In how I design the inclusion and exclusion criteria, there can be a bias in that. So let's say I want to exclude all patients who have had more than 5 lines of therapy, right? There's a bias in that. And so randomization, while it is a gold standard, doesn't remove all bias, but we do our best to remove the unintentional biases in clinical trial. Biomarkers. Biomarkers are a very hot topic because of how much we are learning about tumor individuality, or what we call precision medicine now. Biomarkers are inclusive in almost all clinical trials now because we want to have that one specific thing that we find for you as the individual to direct your therapy. So most of us in this room know about CA125, right? It's a great biomarker for ovarian cancer for many people, not all. but it's not the best biomarker for everything. And so we incorporate new and novel biomarkers into many clinical trials, but there's different ways to think about these. There are prognostic biomarkers, and so these are things like your tumor stage or your grade or the outcome from your surgery. There are predictive tumor markers to consider. So what's the response to certain receptors like a progesterone receptor or estrogen receptor? There's integral biomarkers. So these are required for clinical trials. So let's say we were designing a trial that required you to have a BRCA mutation. That would be an integral biomarker that was necessary. Integrated would be one that we're looking to evaluate but isn't required. So let's say we designed a trial that we want to see if BRCA positive patients responded better, then it would be integrated into the trial, but it wouldn't be required for the trial. And then there's exploratory, and those are used to help develop new hypotheses. So nothing about your treatment on that trial is affected by those. They're really just hypothesis-driving therapies or tests. And so this gets into really precision medicine. So these are classifications of different trials that we consider umbrella trials versus basket trials. This is what we think about as the marker for precision medicine. In umbrella trials, we have one tumor type, but we evaluate to see what the different genetic mutations are in that, and we offer you different drugs based on those mutations. The opposite of that would be a basket trial, is everybody's got a one single mutation, but can have any type of cancer, and we're testing that drug on that mutation. And so this is really how we're trying to develop precision medicine these days. And these are different approaches based on different mutations and tumor types. But this is the way a lot of us are thinking about in precision medicine these days. Moving on to trial assessment. So once you're on a clinical trial, how do we assess how you're doing on the clinical trial and how the clinical trial is doing? So the two big ways that I'll talk about today are, one is imaging, and the other is how are you doing? What are the side effects of this? And those are what we call adverse events. RECIST and CTCAE are the real mechanisms that we use to assess these aspects. And this is how we really look at these specific aspects. So I'm not going to get into the specific of what RECIST measurements are, but what I want you to take home from this is that we have a set criteria that we've determined to determine what your cancer is doing from a standpoint of, is it growing a little bit? Is it growing a lot? Is this therapy working? And we look at a number of different lesions. We measure them in different ways. And this helps us give objectivity to something that can be very subjective from CAT scans or PET scans or MRI or something along that lines. And ultimately, we use that to go into the classifications of complete response, meaning all the tumor has gone away, partial response, meaning the tumor has shrunk a certain percentage, or progressive disease, meaning the cancer has grown a certain percentage, and therefore we've determined that this therapy is not working. And then there's stable disease, meaning somewhere in the middle that we think that this therapy is keeping everything from growing. It may be growing or shrinking a little bit, but overall you're getting benefit from this therapy. There's things like non-target lesions. Again, I'm not going to go into the details of those. With immune therapy, there are a new classification of how we think about things because of how the immune system makes tumors sometimes grow a little bit before they shrink, and it allows us a more 3D model of everything. But just know that this is kind of a moving target in how we are assessing clinical trial imaging because of the moving target of therapies that we're offering these days. Adverse events are the fancy way of saying, what are the side effects of this drug or just in general life? And ultimately, it's our job as providers to really look at every single thing that's happening to you as the individual in this trial. And we'll even ask as simple as, do you have a hangnail on this trial? And then if you tell me in clinic that I have a hangnail today, I then have to determine whether that's related to the drug or not related to the drug. So, I always want to know how you're doing, but it's quite thorough about how much detail goes into making sure that these drugs are safe. And so an example of this is that we actually provide objectivity to many subjective things. So very simplistically, the easiest one to think about is anemia, or when someone has low hemoglobin. This is objective already as it is, right? It's a set number. And so we put in a certain grade. Everything is graded 1 through 5. 5 is always death, and 1 is always, it's noticeable, but has no effect on your daily life. And then there becomes a whole subjective range in between there. And I use this as an example because this is easy, but you can even get finite into, if you look at grade 3, transfusion indicated, hemoglobin less than 8. Well, let's say your hemoglobin was 8.2, and then, but you and I both felt that you needed a blood transfusion to make you feel better. So is that grade 3 or is that grade 2 because your hemoglobin was greater than 8? And you could live without a transfusion, but it would make you feel better if you had one. So there gets to be all these fine, picky things that we talk about to do all this. Ultimately, these get lumped into the entire trial, and at every clinical trial or presentation or publication, you'll see a table that has all the side effects. And the ones that we typically segregate out are the grade 3 and grade 4, because those are clinically relevant side effects, meaning that they required a hospitalization or some sort of treatment. Whereas grade 1 and grade 2 are, I've noticed that the neuropathy is getting a little bit worse, but it's not really bothering my day-to-day life, and I just want to let you know about it. And so these are the things that we take into consideration, and therefore can advise future people of, well, when you get this therapy, when it's approved, this is what you can expect, that 50% of people had this side effect and 70% of people had this side effect. And so that's important to recognize. Trial endpoints is another interesting thing to think about how we design trials, because we have to have an endpoint to any trial. And how we decide that endpoint really helps to determine whether we end up with a positive trial or a non-influential trial. And these are some of the terms that we consider when thinking of endpoints and how to power our study statistically. So the simplest ones to think about are response rate is based on the imaging, our tumor shrinking, like we talked about before, or staying the same. Time to progression is, as you can imagine, just time to the disease progresses. It's similar to progression-free survival, and that's the most common one we use these days. Progression-free survival is the time from entry into a trial to the disease progressing, death, or loss to follow-up. The things about these types of trials is they typically require blinding, oftentimes have a placebo. They have to have some sort of control design. But this is beneficial because we can get quicker results of the trial to prove that this therapy is working. Now, the gold standard that we all want is curing, right? We want to cure this disease. And overall survival, we all recognize, should be the best and only trial endpoint. Unfortunately, it can't be an endpoint for every trial, or the primary endpoint for every trial, because of its limitations. And those limitations are things to think about, like it would take years and years on some trials for us to get results, to be able to get that drug approved, to be able to benefit people off of trial. The other thing is that it doesn't take into a factor subsequent therapies. So let's say you were on a trial and it stopped working after a year, and then you went on another trial or just another standard therapy that worked exponentially better for you, and that improved your survival. Well, the first trial is still monitoring your overall survival, and therefore the results are skewed because of a therapy that you received after that clinical trial and weren't necessarily related to the trial drug that you received on that first trial. And then another great outcome that we all like to incorporate is what we call patient-recorded outcomes, or you may see them as PROs. Everybody loves these in a sense that because it gives us real world data of how are you as the individual patient affected by this trial? How are the side effects affecting you? How are the clinic visits affecting you? How is everything affecting you? This goes beyond the benefit of just what is your tumor doing. It's inclusive of how are you doing, because as many of us know, the reality is even if we can shrink the tumor, we can make your life miserable with some of these therapies, and that's not any of our goals. So patient-reported outcomes are often very important. They're sometimes the bane of yours and our existence because they're like 30 to 40 page forms that we ask you to fill out all the time, but they are very important.
Dr. Corr, we have a question that I wonder if I could pop in and ask.
Please.
Somebody's curious about whether clinical trials would be recommended for clinical trial through their oncologist, which I believe is true, or are our names and conditions somewhere in a database for trial study directors to look at?
Great question. Your names are not in a database. We would love to be able to just pull up a database and say, hey, I've got this new trial. I think you would really benefit from it. Unfortunately, those don't exist. There's a whole number of reasons behind that from a HIPAA standpoint and patient safety standpoint. There are sometimes, there are ways to offer trial patients based on new genetic mutations. So an example is there are some genetic analyses companies that keep historical data, and some institutions will be able to run that data and say, hey, I've got this new trial that's based on this gene mutation. Are there any patients in our hospital that have had this gene mutation? And there are ways to do that, but then that information then goes to the provider and so that provider then can say, oh yes, this patient may benefit from this trial, I'll offer it to them, or they may say this patient's not eligible for this trial, and so it doesn't get directly to the patient. And those are pretty uncommon. The number one way to find out about eligibility is talk to your provider and say flat out, are there any clinical trials that you have available here? And the follow-up question is, are there any trials that aren't available here that I should be considering? And that information is available to you on something called clinicaltrials.gov, and I have it listed at the end of this. And that is a publicly domain of all clinical trials being run in the United States. So when I open up a trial, I have to register it with clinicaltrials.gov. And there's a search form in there to search by ovarian cancer, by certain type of ovarian cancer. But what the limitations of that are, you may get a list of certain trials, and it'll actually tell you what institutions have that available. So if it's not the University of Colorado, you don't want to find out that the University of Maine has this trial open, and you have to travel to Maine to get on this trial. But it'll tell you the sites. What it doesn't tell you is that trial actively enrolling. It always has that little enrolling on the top right thing, but sometimes because of the way trials are designed, they may not be enrolling currently, or they may have a halt on enrollment, or a certain site might not have certain slots for that. So it's always your benefit to reach out to them, and there are contacts there to say, is this trial really open for, and would I be eligible? And those connections, then there's contacts on those web pages to get that information. But the number one thing is to talk to your provider. And some of you on this call know that I will talk to you even about trials that aren't included at our institution. It's quite frequent that someone will email me or call me and say, I've found these 3 trials. Do you think any of these would benefit me? And I'm able to kind of help weed that out and say, this trial A and trial B, A and B, while you may be eligible, don't really make a whole lot of sense for you. But trial C, while it's held at MD Anderson, may be a really good option for you. And I think that would, be something you should entertain. And so that's quite common to do.
I'm just going to pop in one more time and mention, for those of you who don't know, that the Clarity Foundation also has a great clinical trial finder, and they actually have people who can help navigate that and help you understand that as well. But of course, the bottom line is to talk to your doctor. So thank you.
Yeah. So the other thing about trial endpoints, before I move on, is also, as I said, we tailor trials to what the endpoints are, but the FDA also really tailors to what we approve on. We don't basically just approve because it showed benefit in one area. So typically we need benefit in multiple areas. So overall survival, yes, we'll get blanket approval for, because if we cure more patients, who's going to not approve that therapy for whatever reasons. But if you just have progression-free survival benefit of 2 months, the FDA may decide that's not beneficial to approve this therapy. We may need to consider how are patients doing on it, what are the response rates, what are the patient-reported outcomes, and so it gets beyond just having one endpoint being beneficial. The conclusions of today that I really want to emphasize are kind of what we just the topic that we just talked about is that clinical trials are not out there to just improve the science. Absolutely, us as the clinicians want to move the needle forward and improve outcomes for all women, both prevent cancer from ever occurring, from improving your quality of life, from keeping your disease away as long as humanly possible, and hopefully getting rid of it completely. That's the ultimate goal for all of us, for us, industry, the government. But the way we do that is through some sort of objective manner, and this clinical trial fashion is how we do that. I think clinical trial options are always excellent to offer people. They're not always right for everyone. So, in consideration beyond of, is this trial the right thing for your disease? Is it the right thing for you? So if you have to travel to Texas for a trial, is that the right thing for you? For some people, the answer is yes. For some people, the answer is no. Do you have to be in the hospital 5 out of 7 days for this trial? Is that right for you? Is that not right for you? It's more than just, is the trial available to you? Is it the right thing for you? Your resources, we just went over that, in detail. So please, I encourage you to always ask. And if you're ever getting an answer that sounds fishy to you, ask someone else. In my opinion, second opinions are never a bad thing. No provider should ever take fault or criticism or anything about someone getting a second opinion. We do it all the time. I know for a fact, if someone comes to me, a patient, even who I've seen for years, and says, I'd like a second opinion, I'm encouraging that, because if someone has something that I can't offer you, great, if it benefits you. It really should be a strength to you as your own advocate, or your caregivers as your advocate. It's important to seek these options out. And clinical trial is not always the right answer for someone, even if it's available at this institution. It's not uncommon for me to say, hey, I've got this clinical trial available, but it doesn't make a lot of sense for you to go on this right now. The standard therapy, I think, is going to be better for you, whether that's from an outcome standpoint or a tolerability standpoint. I have that conversation frequently too, to say, we can do this trial, but this therapy might be better for you right now. And the other thing is, clinical trials are always evolving. If there's not something available to you now, in 3 months, we may have something available. So check back in with you and continually ask your providers, hey, is there something available now? The best times to ask about this are when your previous therapy has stopped working. So if you're on a new therapy and that therapy is working, or we don't know if it's working, we're very rarely going to take you off that therapy to put you on a clinical trial, because one of the eligibility is typically patient has progressed on the last therapy. And so the times to be thinking about this are when it's time to start thinking about new therapies or maintenance therapies. That's the time to be asking your provider.